Okay, let's get started. It's a great privilege to introduce today Peter Visscher on behalf of Pat Sullivan. Pat couldn't make it today, so he asked me to help out. My name is Roel of UCLA. Peter happens to be a countryman, a Dutch origin, like I am, so it's a even greater privilege to invite him today or host him today. I sometimes wonder if Peter, when he started out doing his studies in agricultural university or college, that he was even thinking about human genetics at the time. But I'm really glad that he made that transition over the years, because if there's any person, he's one of those few handful of people that really have made a big difference in how we consider heritability, and even the term SNP heritability. I'm not sure he coined it, but it was pretty close to what he did. And he's not simply presenting today something that he did in the past, but pushing the field even further in understanding heritability of complex traits. So I'm really glad that Peter, who is down under, as we would say, from the University of Queensland, is joining us today to present his work on whole genome sequence data and the estimation of genetic variance. Peter? Before you start, one more practical point. Peter will present his work, and then afterwards we will have a Q&A. And I ask you all who are listening, to do that through the texting mechanism, either the Q&A button or the chat button, so I can relay the question to Peter and he can answer it. But Peter will first do his presentation, and then we will move on. Peter?
Thank you very much, Roel, for your kind introduction and for chairing this session. As you mentioned, Pat Sullivan, Pat approached me a while ago and said, please, can you give one of these presentations about a preprint that we put on bioRxiv just over 2 months ago. And that preprint is about the estimation of genetic variation from whole genome sequence data. And the focus of my presentation is on what's in that preprint. This work is a large collaboration. It's between a number of authors here at the University of Queensland, in particular Pierre, who's a PhD student of Jiang Yang and myself, our colleagues at the University of Washington in Seattle, and the TopMed Consortium. And just to emphasize that, as usual, these large studies are really a big consortium efforts. I've put here up all the authors and banner authors of the preprint that we put on bioRxiv, and this is a picture of Pierre. So the outline of my presentation is as follows. I want to give just a little bit of background about, to put the presentation in context. Something about genetic architecture and the estimation of additive genetic variance and of heritability, and a very short summary of the inference from genome-wide association studies with SNP arrays to date. And then I'll go on to the actual material in the preprint, the analysis of the TopMed data, where we estimated and partitioned genetic variation using whole genome sequence data. We looked in detail at population stratification and did a little bit on functional annotation. And I would like to end with some conclusions and possible implications of the findings, of our findings. So these may be some very obvious statements, but I'd like to go through them nevertheless, because they put our study in context. The first is that genetic architecture, which we can define as the joint distribution of effect sizes and allele frequencies, that's actually determined by past evolutionary forces, such as mutations, genetic drift, and natural selection, and gene flow, etc. And that the success of genome-wide association studies, at least how they have been conducted to date, which is with SNP arrays, really depend on what the unknown genetic architecture for our traits actually are. And finally, because polygenic risk scores are so much in the news, the precision of polygenic prediction depends on the total amount of genetic variation that's captured from our genomic technology, and how well the SNP effects are estimated. So they also, the precision of polygenic prediction also depends on the genetic architecture. In the past, when we estimated heritability, it'd be done through a pedigree or family or registry study using identity by descent relationships between individuals. And that kind of method is actually blind with respect to genetic architecture. So if the assumptions of the model are correct with pedigree or twin studies, we should be able to estimate the total amount of genetic variation that is in a population. And of course, with GWAS data, that is not the same because it depends, our analysis, our estimation of genetic variation from common SNPs depends really how much genetic variance is actually captured by those SNPs. And that depends on the linkage disequilibrium, LD, between the unknown causal variance and the SNPs we have assayed or imputed. We can actually make some predictions on genetic architecture just from using some theoretical models. And I'll just show you two extreme models here. These are just from theory. And the left one is from a simple neutral model where the trait that we're considering does not have any correlation with reproductive fitness. And I've also assumed in this simple plot, which is actually published in Molecular Psychiatry, this plot, a constant effective population size. This simple model makes already quite also a simple prediction that we can test empirically, because this simple model would first of all suggest that most of the causal variants are actually rare. This is the cumulative proportion genetic variation, but that the variation, the genetic variation contributed by the causal variance is linear. The cumulative variance is linear in the minor allele frequency, which is here on the x-axis. What does that mean? That means that under this simple model, for example, 20% of heritability would be contributed by causal variance with a minor allele frequency less than 10%. Yeah, that's this linearity. And essentially what natural selection does, if our trait is correlated with fitness, is put this line on the diagonal towards the left, which means that a disproportionate amount of genetic variation will be captured by rare variants. And there are many models of relationship between reproductive fitness and our trait that one can come up with. And I'm just showing you on the right side of this picture on slide 6. I'll show an example from a paper from Adam and Walker from nearly 10 years ago, where he modeled his relationship between the trait and fitness with these parameters here. I don't have details to go into this model, but essentially what it means is the larger this value here, the closer the trait is to reproductive fitness itself. And as you can see, I've just got the cumulative proportion of genetic variance again on the y-axis, that now we've deviated from this line y equals x. So the closer the trait is to fitness, the more genetic variation is captured from rare variants. So this kind of model, selection model, predicts that more than 20% of the heritability would be captured from causal variants with a frequency less than 10%. And recently, using SNP data, a number of authors have suggested we can model that and in a simple model try to empirically look at the relationship between allele frequency and effect size. Here is a very simple model where we have our effect size on our trait being a function of allele frequency, to be more precise, heterozygosity at that marker, and a parameter here which we called S, which has to do with the relationship between the effect size and the allele frequency. And this model, so the effect sizes depend on the frequency and on this parameter S, and that means that the variation, the genetic variation contributed by this particular variant, the heritability due to that variant, is a function of heterozygosity again, and then the square, the variance of the effects, and that comes out to be like this over here. And you may say, well, you know, what can I do with that? But this simple model already gives two nice contrasts. For example, if this parameter S is equals 0, yeah, that means that that common variants explain most of the heritability, most of the genetic variation. It's basically the same as the neutral model I went through before, whereas S equals -1, yeah, you can see this term drops out. That means that all variants, irrespective of their allele frequency, explain the same amount of variation. Or if you wish, rare variants have bigger effects. That is S equals -1. So a number of authors have suggested that we can model GWAS data like this and actually estimate this parameter, S. In a paper from Jiang Yang's group that was published last year in Nature Genetics, we attempted to do that using data from the UK Biobank. And here are just the results of this parameter S for a whole bunch of traits. And without going into great detail, the average across many traits is roughly -0.5. Now, this already leads to some interesting implications, and I'll just go through this as a complete aside to the rest of the talk, which is, what if we were to conduct GWAS not with SNP or imputed SNP data, but using whole genome sequence data? Then this relationship we and others have inferred about the association between the effect size and the frequency makes a prediction about the power of detection of a rare variant compared to the power of detection of a common variant. And although rare variants have bigger effects, they're not, the relationship is not so strong that it compensates for the fact that those variants are rare in the population. And it's the combination of frequency and effect size that determines statistical power. So everything else being the same, it actually means that to detect, relative to a common variant, to detect a rare variant with a frequency of 1%, for example, we would need 5 times the sample size compared to the common variant, and even more disproportionate, larger sample size for very rare variants. This, of course, is very simple. This is assuming everything else is being the same. We just do exactly the same for a GWAS as we do now with SNPs, as we would with whole genome sequence data. So this is just a reminder of the power. So here's my summary of where we are with genetic architecture for a model trait human height. And this, to my knowledge, is the most up-to-date one. So first of all, pedigree and other studies suggest that by and large, the heritability of height is about 80%. When we look at the genome-wide significant variants detected today, these are the robustly associated variants for height, or just over 3,000 of those in the publication last year in Human Molecular Genetics. And they together explain roughly 20% of the total of total phenotypic variation, so a quarter of the genetic variance. About 10 years ago, we first estimated SNP heritability using genotyped SNPs only. That was about 300,000 SNPs or so, and we estimated that 45% of phenotypic variance was captured by those common SNP variants. And more recently, we and others published that number went up to 56% if we use SNPs imputed to a fully sequenced reference panel. So that's the situation with height, and I don't have a picture of what it is for schizophrenia, but you will have or other psychiatric traits, but you will have those numbers yourself. If I try to summarize it for a few other traits, so here we have the results for height again, here for the results for body mass index. Pedigree studies vary a bit more widely, but estimates of heritability 40 to 60%. We and others first looked at this in 2011, I think, and estimated 11% captured from genotyped SNPs, and more recently 27% from imputed SNPs imputed to a fully sequence reference panel. And really the question here is, what will we estimate if we go from imputed data to a whole genome sequence? How much larger essentially will this number get? And in particular, are we going to recover these estimates at the top here, which are the full heritability due to segregating variants in the population? I have attempted here to give you results for educational attainment and schizophrenia as well. These are my guesstimates, and you can tell me whether these numbers are appropriate for schizophrenia, for example. So the rest of the talk is about our preprint now. So this was all background. With the collaboration with the TopMed consortium, we got access to data on nearly 40,000 whole genome sequences. And those individuals all had a measure, they were all adults and had a measure on height and body mass index. And formally, the sequence data is from the TopMed data freeze 5B. Our colleagues at the University of Washington had already done a lot of QC on the phenotypes, and we did some extra ones where we basically removed the effects of sex, age, and specific study for mean effects or variance effects. And I should have said these data are from, I think, 16 different studies that are participating in the TopMed consortium. This looks quite messy, and it was kind of as anticipated. This is a principal component plot from the 40,000 samples that we received using only common SNP variants to do the PCA. So this is on about 1 million HapMap3 SNPs. And the ones, I hope you can see the colors, the TopMed samples are in purple, and they're mixed in with the HapMap samples here. And you know, we knew there were a lot of ancestral differences among the TopMed consortium study cohorts. We were particularly interested in answering just one question, which was, if we have a sample like we and others have used in the past of European ancestry, how much genetic variation can we capture using whole genome sequence data? So after a lot of anguish, really, we had to throw away nearly half of these data to end up with a sample of just under 22,000 unrelated individuals with European ancestry. And after the QC of the sequence data, and again, I don't really have time to go into detail into this, we ended up with just under 48 million sequence variants. The estimation procedures are, so at the end of this QC, we had just shy of 22,000 samples, phenotype, height, and body mass index, and 48, just under 48 million variants. The analysis we did, and I'm going to show you the results of, are all of a method that we published a number of years ago, which is by annotating the SNPs according to frequency and LD, and then estimating genetic variants within each of those bins, and then adding it up to get a total. So that's the partitioning here. So formally speaking, it's a linear model shown over here, where the variance components, these are the random effects here, and the covariance structure of those is that we have a genomic relationship matrix for each of the SNPs in a particular group. And we can partition this as much as we like, although the more we partition, the larger the standard errors of the individual variance components will become. So this method, we named it LDMS for LD and mass stratified estimation of genetic variance. And that was published in a paper in 2015. for most of the analysis, so the default analysis for which I'm going to show you the results, the partitioning is by 14 bins. So we annotate a SNP according to its minor allele frequency and whether its LD score is high or low. So this is a busy slide, but it's an important one, so I'll try to take you through what all this means. First of all, the results for height are on the left-hand side, and the results for BMI are on the right-hand side. This initial analysis is just a check that a quality of our sequence variants and the quality of our phenotypes were as we would hope them to be. So what we did here is we took all our 48 million SNPs and got rid of 99% or so of them until we only had variants left that were on any of these three arrays. So then we basically mimicked here as if we had a SNP chip experiment and not an analysis based on whole genome sequence data. So after we had retained the SNPs on any of these three arrays, so the axiom, the GSA array, and the human core array, we then did the standard imputation analysis using not the TopMet as a reference, but using the HRC as a reference panel. So we basically mimicked the standard SNP chip plus imputation procedure here. And this is because we had a benchmark here from previous publications on what estimates to expect. After we did that, we estimated the variance components using our partitioned analysis. So here on the right-hand side, I have the variance components for the low LD bins, and on the left-hand side for the high LD bins. So there are 14 bins in total here. These are the estimates with their standard errors on the x-axis here, and here's the total by summing up all these variance components. And the total, depending on the array, was about 60, just over 60% for height, and about 25–26% for BMI. And remember, when we did this before on SNP data, our estimates were 56%, I think, and 27%. So these results are quite, given the standard errors, also quite similar to what we estimated before. So this gave us confidence that in the quality of the data, that if we just picked out the SNPs, they behaved like any study where we take a sample of individuals from the European population and estimate a SNP heritability after imputation. So we next said, okay, the quality of the data is good. Let's do exactly the same, but throw in all the 48 million sequence variants and do the same partitioning. And that's what's shown over here. So first of all, the most exciting results here are the totals, which are just under 80% for height and 40% for BMI, which are very close to what we think the pedigree estimates are. So if our inference from this analysis is correct, we have indeed recovered the pedigree heritability using whole genome sequence data. There's more interesting, there's more interesting things to point out on this slide of results though. By the way, this is an analysis on the 22,000 where we only fitted 20 PCs, and I'll come back to that a little bit later. And these are PCs generated from common SNPs. First of all, what you can see here, in particular for height, but also for BMI, is an apparent enrichment of genetic variation of SNPs in the low LD bins. And what you can also see, despite the large standard errors, there seems to be an enrichment of genetic variation here in SNPs that are both in low LD with other SNPs nearby, and also have lower frequency, less than, say, 10% or so. Same for height and for BMI. One of our concerns was that the annotation that we used in the previous set of results, the annotation of minor allele frequency and of LD, was from the data itself, was from TopMed. So we wanted to check what would happen if we used annotation from a completely different sample. So we went to the sample of the UK 10K, and we looked for the total number of sequence variants that overlapped. We found 14 million variants that overlapped, so a substantial smaller number of variants that overlap between the two sets of sequenced samples. And we used these 14 million variants, the annotation of MAF and LD in the UK 10K, to put our SNPs into bins and do the same estimates. Now, the crucial thing about this slide is not the actual amount of variation that we explain in total, which, as you can see here, is less than when we use the TopMed data, because we only have 40 million variants here. The real comparison here is between the left and the right, where we have TopMed annotation versus UK 10K annotation for the SNPs. And as you can see, the results are extremely similar, both for height and for BMI. So from this, we concluded that the annotation that we've used, which was specific to the TopMed data, did not unduly influence our results. The next question, and some would say the elephant in the room, is population stratification. Why is this important, and in particularly important for rare variants? In a paper in 2012, Ian Mathieson and Gil McVean pointed out that rare and common variants lead to a different confounding, whether we're looking at rare or common variants in a population. And they concluded here in their abstract, we demonstrate that rare variants can show a stratification that is systematically different from and typically stronger than common variants. And this is not necessarily corrected by existing method. So this can be potentially serious if we're dealing with a spatially structured population where there are also differences, phenotypic differences between the spatially structured groups. Another example, a more recent one, is from a number of authors who posted 2 papers on bioRxiv, essentially the same set of authors. And this is a formidable group of authors, so I'm not having a go at these authors, just showing you how difficult it is to distinguish genetic variation from rare variant confounding. In the first paper, the authors used a clever way to try to also get at rare variants using haplotypes of SNPs that were derived from common from arrays and from imputed SNPs, and they're called these haplo-SNPs. And they concluded by constructing genetic relationship matrices from these haplotypes that they could recover nearly the full heritability for a number of diseases, including for schizophrenia. After a lot more subsequent analysis and simulations and modeling, the authors actually in a second paper concluded that this was a bias. The estimates were biased upwards due to subtle stratification. And they showed in this particular second bioRxiv paper that constructing principal components from their haplotypes reduced the effect of stratification, but they concluded here in the end, which is a similar conclusion to this one at the top here from Mathieson and McVean. Overall, our results suggest that estimating the heritability explained by rare variants for case control traits requires exquisite control for population stratification. But current methods may not provide this level of control. And to my knowledge, neither of these papers have been published subsequently in a peer-reviewed paper, but they're a very important reminder of the possible effect of subtle population stratification. So what did we do to look at that? Well, the first thing we did was just quantify how much variation in our TopMed sample is actually explained by principal component and principal components. So this is just a simple linear regression of our phenotypes on a number of PCs all the way up to 760, which include PCs based upon very rare variants. The top results are here for height and the bottom ones for BMI. And for each of the traits, we have either the trait itself or the rank-based inverse normal transformation, so transformed value of the traits. And you can see for height, if we fit 20 PCs, about 2.5% of variation explained, and this goes up to just over 3%. And for BMI, it starts at about 0.25% or so and goes up to 0.5%. Now, these results here are actually quite similar if you were to take a sample with European ancestry from a sample from North America, where we've looked in the past, where we explain about 2 to 3% of phenotypic variation from PCs. So we were not really unduly worried about these results. And there's also, I think, after this initial increase, we see this sort of steady state, slow increase, which would be just consistent by fitting more and more covariates. We expect this number to go up the more variants we fit, and we fit up to nearly 1,000 here out of our 40,000 20,000 samples. We next looked at the effect of actually just including more PCs in our model. Yeah, so here we have where we fitted either our original 20 PCs, and we go up to 280 PCs fitted in a model of analysis. And that will be, the 280 would be 20 PCs in each, from SNPs in each of these 14 bins. And I hope you agree with me that the results changed actually very little, and we still end up with a total estimate of heritability of 75 to 79% for height and about 40% for body mass index. So from this, we concluded that as far as we could see, by trying to model those rare variants or account for population structure as best as we could, that there was no strong effect on the estimate or the partitioning of our variation. We went recently even further. This is not in the preprint, where we fitted up to 100 PCs in the rare bins. So the rare bins now, these rare bins over here contain 100 PCs. And again, the results did not change appreciably, depending even if we fit 760 PCs. So again, our conclusion from this was that it appears that we have adjusted for subtle population stratification appropriately. We were still not 100% convinced, so we did an additional analysis. This is again not in the preprint, where we took advantage of the recent availability of exome sequence data in the UK Biobank. So this is clearly not whole genome sequence data, but it's still useful because the UK Biobank contains spatial information where people were tested and where people were born. So PIRIC extracted in total just under 36,000 individuals, unrelated individuals of British ancestry, where we had height and BMI. We had spatial information, either the assessment center or coordinates for the place of birth. And we had genotypes or sequence variants, in total 2 million. Of course, this is a lot less than we have from the TopMed sample, because this is only the exome plus some common variants. And here are the results for estimates of total heritability from UK Biobank, where we either made no adjustment for spatial information or we adjust it for birth coordinates in green, or in red for the center of assessment. Results for height and for the transformed height at the top, and for BMI and transformed BMI at the top. For height, I hope you agree with me, very little difference between these two ways of adjustments. And for BMI, there is a small, a difference here between no adjustment and an adjustment. Not sure exactly why this is, whether there's really some subtle stratification in the UK, depending on where you're born, and body mass index, for example, some environmental effect or so. But for height, which of course is a very ancestrally stratified trait, very little difference. So from this, we concluded that at least for the UK Biobank, and taking account of spatial information, that doesn't seem to be a big effect thereof when you fit it in the model of analysis. Finally, we went back then to the data from TopMed, and we, with help from our colleagues at University of Washington, we used some functional annotation. Because we had prior information that those SNPs that were particularly at low frequencies and were in low LD contributed a disproportionate amount of variation, we further partitioned those SNPs into whether the variant was protein altering, so this would be missense, loss of function, and non-synonymous mutations in general, whether they were coding variants but non-protein altering, or whether they were just any old variant that were not annotated to change proteins directly. And we estimated a lot more variance components, 20 now, so we're pushing it given the sample size that we had. But when we did that, these are the results. The totals again are similar to before, just under 80% for height, and we actually increased our variance estimate of variance a bit for BMI, but look, there's a large standard error here. But interestingly, so you could say, well, this looks essentially no different to before, or what could I conclude from that? What's crucial is that some of these bins have a very different number of genetic variants in them. For example, there's relatively few coding variants that change a protein in these data. So what we did, and that's shown on the right-hand side, is take these variance components and divide them by the number of variants in each of these groups. So that is essentially an estimate of the effect size, an average kind of effect size or enrichment of the effect size of different annotations. And what's shown on the right-hand side, although the standard errors, as you can see, are huge, is there seems to be a strong enrichment of rare variants that are protein changing, that have the biggest effect. Yeah, that's the variance components divided by the number of variants in that bin. So we concluded from this that this is evidence for larger effect sizes, essentially, of low MAF and low LD protein altering variants. So finally then, to come back to where we started with, which was talking about genetic architecture and plotting the cumulative contribution of genetic variance as a function of minor allele frequency, we end up with this picture over here on slide 25. And that is, if we're looking here on the y-axis, and we're looking at the 10% again, that we found that, given the standard errors here, that about 40 to 50% of total heritability is contributed by variants with a frequency of less than 10%. And remember, under a neutral model, the expectation here would be 20%. So we clearly have a strong enrichment of genetic variation among the low frequency variants. We also try to quantify this for going further than 10%, and I'll just show you the results here for what the cumulative proportion is for 1%. That's not that's 90% for height and 16% for BMI. But look at the standard errors here are actually very, very large. So I'm reluctant to draw strong conclusions about these estimates over here. So in conclusion, then we concluded from the evidence, from the data and the analysis we performed, that indeed we seem to have recovered close to the pedigree heritability from whole genome sequence data on unrelated individuals. We find, as we and others found before, strong evidence for negative selection, meaning that variants at lower frequency on average cumulatively explain more variation and have bigger effect sizes. We find an enrichment of genetic variants among rare variants in low LD with other variants. We find evidence for, on average, larger effect sizes of coding variants. And a big caveat here is that the standard errors of the estimates are not small. Ideally, we would have liked to do this analysis on about twice the sample size, but this is of course already a quite formidable data set. If we can draw these conclusions or extrapolate from them to other traits, from height and BMI to other traits, including common disease, then of course this has some potentially big implications. And the first one is that, well, obviously it leads to, what experimental design do we need to detect those variants if we want to detect those individually. But it also has implications, for example, for polygenic risk scores, because we know that the upper limit of a polygenic risk score, the accuracy we could ever get, be larger in principle with whole genome sequence data than with SNP array data. Because with the whole genome sequence data, we would just capture more genetic variation. However, and this is a big however, as I showed before, we need disproportionately larger sample sizes to estimate the SNP effects accurately for rare variants, or even to detect them individually compared to common variants. And I think there are plans to perform whole genome sequence analysis on the entire UK biobank, and that will be a really good test case unless there's other huge samples that become available sooner. All our analysis have been done on quantitative traits, and the reason for that is that we could get a sufficiently large sample there. Of course, it's interesting to think about, to do these kind of analysis on case control data. And I think there are some extra challenges there. As others have pointed out in the past, these are also challenges that occur with just SNP chip data. We can have batch effects correlated with case control status, but population stratification in particular may be very hard to deal with, as I pointed out in some of these papers from Alcus Price and colleagues. For example, we could have that from an individual cohort that contributes to a larger sample. We could have that cases and controls are not spatially or ancestrally sufficiently matched. Or we could have a number of unbalanced case control studies that come together where different individual cohorts are not spatially or ancestrally matched. And I think be hard to deal with that. We have not tried to do this with disease data as yet. And I think there's also some issues when we go to the analysis of 0-1 data using a linear model, as we've done in the past, in particular for rare variants. So more work to be done there. Now I've got everybody on the line. I'd like to finish by saying you should all come to Brisbane next year, where we are hosting the International Congress of Quantitative Genetics. And the chair of this meeting is Naomi Ray. And it's be a wonderful conference that has a fair amount of human and and sort of cellular and other model organisms in it, but also you'd find out what's happening in other species and also theoretical developments. So please come to sunny Brisbane next in a year from now. And finally, I'd like to thank all the wonderful group of people that I have the pleasure of working with on a daily basis. And I'll end there and take questions, and Roel is gonna chair that, I think.
Yeah, thank you very much, Peter. Thank you very much. Yeah, the paper is now open for questions, and I see already two chats coming in. And I will read the first one. It says this: the results suggest selection pressures on height and BMI. I realize that this question can perhaps only be answered with speculation, but what kind of selective forces and when do you think have influenced the genetic architecture of height and BMI in this way?
Well, thank you for that question. I mean, the evidence for selection is on the variants that are associated with these traits. We can't even say for sure whether selection itself has been on height or BMI, but there is evidence for those variants associated with those traits. What they may be, I'm not sure, but there is evidence for contemporary data that there is a small correlation between how many children you have, for example, and how tall you are. In the contemporary UK Biobank data, there is a small correlation there, which is in a direction that would predict that over time you get small differences directional differences in height. And also there is some evidence for stabilizing selection that's saying individuals who are too extreme tend to have a lower fitness. I think also to point out that the evidence on selection has been made before by a number of groups, so the evidence therefore has been presented. What we're showing here is just essentially more of it and quantified better, probably because we have sequence data where we can get at the sort of cumulative association with the traits a bit better. But thank you for the question.
Yeah, Abdel was asking the question, and he also said this: "Oh, thanks for the wonderful presentation." That I want to relate it to you. I don't see other questions at this time, so I may take the opportunity, since I'm speaking anyway, to ask you a question. When I look at the data and the results, Peter, they are wonderful in many ways, and perhaps too wonderful, too good to be true. And so that makes me nervous. What is going on? If you can explain all of the heritability that is predicted in family studies, and family studies tend to I would say estimate the heritability because of shared environment and other factors perhaps. So I just wonder if you have any comments there. Can this be too good to be true?
Well, that's a very good question, Roel. Obviously a question we asked ourselves as well. The first thing to point out is that the standard errors of our estimates are of the order of 0.08. So, you know, with more data, these results could change a little bit. Secondly, the estimates for pedigree studies are all a little bit higher, in particular for BMI. I mean, if you take a twin study for BMI, you get a much higher estimate of the heritability than 40%. So it's not one-to-one there. There are also subtle other effects that we know have an effect on either the estimate from family or twin study, or from the estimate from SNP or in this case sequence variance. For example, for height, we know that there is assortative mating. So this then has an effect on genetic variation, and it depends on whether you estimate that genetic variation assuming there is no assortative mating, or you just take the genetic variation in the population now, as you do in a twin study, for example. just to contrast some of these estimates, in a paper from Alexander Young and colleagues last year, I think, in genetics, they used data from Decode where they tried to use only the segregation within families to estimate genetic variance. And they concluded from their data that the heritability for height was less than 60%. The total heritability for height was less than 60%. This is in a randomly mating population, so not including assortative mating. So there are all these kinds of effects, and I think just larger sample sizes and like here with whole genome sequence data, or larger sample sizes with family data, where we can exploit segregation within families, will provide the sort of final answers kind of on these questions. So I'm not, I think these estimates are close to the pedigree heritabilities. I wouldn't want to go as far as to say there's no more research required. They are imprecise, and our variance and our estimates, plus those from family studies, may contain biases. I would argue, by and large, there will be biases, but I haven't seen much evidence to suggest that those biases are very large.
Okay, I got another question here from Michael Neal. He says, height is much more stable across adulthood than its BMI. To what extent do you think age-related variation in gene expression is attenuated Gremel estimates relative to those of twin studies?
Yes, that's a good question. So if I can rephrase that question, if we take a trait, well, even like BMI, is BMI, when measured age 20, is it genetically the same trait as BMI as if we measured it at 70 years of age, for example? Because when we have samples like this from the population, the age of the individuals ranges widely. My answer to that question is when others and we have tried to look at this in the past, for example, by estimating genetic correlations between these traits early and late in life, we find that by and large, these genetic correlations are actually quite high. And that doesn't seem to be a lot of the variation in the population that is explained interactions between genotype and age, for example. But this, of course, may be a trait-dependent kind of thing. So there may be other traits where things early in life, what seems to be the same trait is actually genetically quite different from later in life. So you really have to model that, and it will have an effect on an estimate of heritability if you lump it all together. Yeah, it's a good question as well. Thank you, Mike.
I'd like to invite other people too to ask questions, and please send in the text. Again, since I'm speaking, I can continue asking additional questions. I noted, Peter, that you gave the example of the UK Biobank, the exome sequence data, in which 60% of the variance was explained instead of the 78. And I thought, well, this is a pretty large fraction if you think that only the exome is only 1 or 2% of the genome. How do you explain the extremely high explained variance in that data set? Or is there something I overlooked here?
Yeah, well, there's not something you overlooked. There's something that I forgot to say, that as part of that analysis, we also fitted HapMap3 SNPs, or common variants, in the same analysis. So we basically have a funny mixture of common SNPs in the whole genome and then exome sequence variants in the exome. Our purpose of using the UK Biobank data was not to make a direct comparison with our estimate from TopMed, but rather it was to see whether there is a large effect of spatial structure and of subtle population stratification. So really the purpose of showing those results was to compare the estimates from no adjustment for spatial structure versus the estimate for adjustment. So I find it hard to compare in the 60 versus 80% when we have a funny mixture of sequence variants in 1 or 1.5% of the genome, and then common variants in the rest of the genome.
Please, more questions are welcome. I see another one coming in. Michael Neal again, you're welcome, he says. Great presentation. It's a relief for the aging twin researcher that biases in twin studies frequently accused without a shred of evidence are likely not at large, at least for these traits. So you've given him confidence, Peter.
Well, I totally agree with that because as he know I would, but if you look at just it's a meta-analysis of twin studies across many traits, you'd find that by and large the identical twin correlation is roughly twice the DZ correlation across many traits. Just looking at the correlations, and you know, it's hard to see for me that that the estimates are systematically biased for all these traits by some kind of funny interplay between common environmental and genetic effects. I mean, you know, the most parsimonious explanation for that observation is that to a first order approximation, you know, most individual differences are simply due to additive genetic variance. But that's a whole different discussion about twin studies.
Okay. Well, we have a few minutes left, again, I can ask another question if you don't mind, Peter, and then if there's no other questions left after me, then we'll close. But what I observed in the patterns in the slides that of the explained variants in the different bins is that the bin that was rare alleles, the rarest alleles, I think, and the low LD was relatively high, explained quite a relatively large fraction of the heritability. And then the next bin of 0.001 to 0.01 explained far less than the third bin. And so that was true for both, for the height and for BMI. And when I see similar patterns in different data sets that I cannot not easily explain myself, I suspect that there's maybe some technical or some other artifact underlying the pattern itself. Have you seen it too? And what is your explanation there?
I don't have an explanation, but I think we should be careful in not over-interpreting the results from individual bins or for comparing them, because as you will see from those results, the standard error of the estimates are quite large. I mean, we haven't done a formal test. My guess is if we did a formal test and on the, you know, differences going up and then down again, and then taking account of all the multiple testing that we do, that it wouldn't be more than expected just by chance. But it's again, it's a cry for that we even for a trait like height and BMI, we need additional more samples to get more accurate estimates of all these particular variance components and of the partitioning.
Okay. Ken Kendler asks this question. Would you predict, Peter, that lower frequency variants will prove more important than we might have predicted for psychiatric disorders?
Well, I don't know. It's a good question, but I find it hard to answer. I mean, the only thing I can say is if you detect rare variants that are robust